All right. This lecture concentrated on RNA, ribonucleic acid. The previous lecture we talked on DNA, deoxyribonucleic acid. Okay, so RNA, ribonucleic acid, is made up of nucleotide containing the sugar ribose. Ribose. It is not deoxyribose. Ribose for RNA. Four nucleotides that make up the RNA molecule have the following bases. So it is A, adenine, uracil, cytosine, and guanine. You notice the difference with the DNA. This one doesn't exist. What is the original one in DNA? Leave it for you. You have to study your previous lecture. Okay. One of the differences is RNA and DNA is the replacement of thiamine with the uracil. You see, this is the different. T in DNA, U in RNA. As with DNA, complementary base pairing, again, they are paired to each other. So cytosine with the guanine, adenine with uracil. In DNA, adenine with thiamine. Okay, another difference is, you remember, double helix DNA. Round each other, two strands. Here it is just one strand. Okay, this is the figure. It is just one strand. Again, it got phosphate and sugar. Sugar is ribose, not deoxyribose. And here is GUAC, and of course at the other side, G combined with C, U with A with U, C with G, and so on. This is a table to compare some differences, main differences between DNA and RNA. DNA present in the nucleus, mainly in the nucleus. Some of the DNA in mitochondria, few in comparison to that in nucleus, while RNA is in nucleus and some we have it free in the cytoplasm. This is DNA carry the genetic material, while the function of RNA to help DNA and its function, transmit information, producing protein, and so on. Here the sugar is deoxyribose. Here is only ribose here. ATCG, as we said, here AUCG, so the difference is U instead of T. Here double strand helix, here is just single one. Here transcribed to give RNA. Okay, copied. Here translated, you got information and you translate it to a solid things, produce protein, produce enzyme, produce hormone. So it is translated. And the message where? The message in the DNA. Where you build the ribosome. Okay. The function. You remember the function, three function of DNA. Go back to your lecture and see the three main function and characters of the RNA. Here, the RNA divided into coding and non-coding. Coding is the messenger RNA. Messenger take the message from the DNA and go to the cytoplasm to produce certain protein. While the other non-coding, like the ribosomal RNA or transfer RNA or the small RNA, three categories. Ribosomal ribosome, yes, forming the ribosome, transfer taking amino acid as a raw material to produce the proteins, and the small RNAs, there are many forms of it and they are controlling the process. Okay, the small RNA are involved in the expression of the gene that code for messenger RNA and RNA. Right, messenger RNA produced in the nucleus where DNA serves as a template How you produce it? You produce it from DNA. Right? This type of RNA carries genetic information from DNA to the ribosomes in the cytoplasm where protein synthesis. It is carrying the message. Carrying the message, therefore we call it messenger. Take message, please, we don't want this type of protein. Messenger linear. Ribosomal. What is ribosomal? In eukaryotes, more than one cell, organism, higher organism like human being. Ribosomal RNA, which is abbreviated like small r RNA, is produced using DNA template and the nucleolus of the nucleus. The structure inside the nucleus, a nucleolus. This is the ribosomal. Then they are joined to proteins, so you have small unit and large unit, and this unit, after attaching to the protein, they either attach themselves to endoplasmic reticulum or stay free. Right. When these large and small unit combine and they make the ribosome, and the ribosome is the factory, is the workbench to manufacture protein. Now the transfer are the cars you are carrying, as if you want to build a building. So you have to carry the bricks, you have windows, glass, doors, everything. This thing has to be carried, and the car here is the transfer RNA. From the name, it is transferring the amino acid to the ribosome to build certain type of protein. The ribosome, the amino acid are bonded together in the correct order. Correct order, of course. If it is not correct, so you are producing something you don't need. Maybe it kill you. So, for example, at this time of coronavirus, your ribosome have to produce antibody against corona, not against anything else, not against cholera virus. No, we don't need it. We need antibody against corona, COVID-19. Who will tell them this type? It is the messenger RNA coming from the nucleus and coming with certain message. Please produce this type of protein. We need it urgently because this patient is having the disease, and his or her cells telling this. This is the immunity. His immunity telling the body, please produce this type of antibody. Okay. We got 20 amino acids. Yeah. They are important person. They can't sit in the car with another passenger. Everyone has a car by itself. So at least we need 20 transfer RNA. Each one take one amino acids. Of course, some of them important that they have more than one car. So you may have more than 20, but you can't have less than this, because each type of transfer RNA carry one amino acids, not another type. So we have at least 20, as I said, maybe more, but not 19. Okay, we have good time, and this is, I think, the last slide. They have nothing to do with the real production. They are not coding. They are controlling. So small nuclear RNA, small nuclear, and here small nucleolar. This is in the nucleolus, here in the nucleus. RNA are involved in the splicing of messenger RNA before it exported from the nucleus to the cytoplasm for the translation. You see, this make it in good shape. When the messenger RNA goes, before it goes for the production, if anything wrong, it will cut it if there is extra. So splicing, cutting, okay? Make it as neat as you need it. Small nucleolar, modify the ribosomal RNA within the nucleus of the cell. So this one working on the messenger RNA, this one work works on ribosomal RNA. Okay. MicroRNA, MI, attached to the messenger RNA in the cytoplasm. Messenger RNA are thus prevented. So you produce antibody, even let's say on the against COVID-19, and the patient survive. There is no symptom. You don't need more antibody. Your body just wasting the amino acids on this expensive material. You don't waste it and you don't need it. Okay, so there are two ways to prevent the messenger RNA from producing more protein. We don't need it. Either to just prevent it, to attach it so that it can't be read, it become blank. They can't read it. On this one, it is naughty. It will kill it. Said, okay, you want to produce more? Please go. So it kill it and degrade it. So all these small RNAs are controlling the process of production. It is not produced itself. This is why they call it non-coding RNAs. I think I have to stop here, and this is the end of the lecture, and thank you.